Now we're going to spend some time reviewing viral genetics. Let's remember, viruses are dynamic entities. They are constantly mutating and adapting, changing their genetic and phenotypic makeup. Step 1 wants you to be familiar with 4 mechanisms by which viruses mix up their genotype and phenotype. So if we think back to genetics for a minute, do we remember the difference between genotype and phenotype? Remember, a genotype discusses or describes the DNA makeup of an organism, while the phenotype describes the physical presentation of the organism, or the way the outside world sees it. The genotype usually determines the phenotype, but as we will see, that is not always the case. If we remember back to meiosis, during which phase do homologous chromosomes line up and exchange genetic material? That would be during metaphase 1. The homologous chromosomes shimmy up next to each other and exchange genetic material and recombine in a process called homologous recombination. Viral genomes are capable of doing similar things. If they have similarity, they can shimmy up next to each other, attracted to each other by the regions of homology, and exchange genetic material. Here we can see a picture of chromosomes in metaphase 1 of meiosis. The sister chromosomes, the homologous chromosomes, line up together next to each other. They cross over and then split, forming unique sister chromatids, or recombinant chromatids. The same sort of mechanism happens in virology. The DNA or RNA is literally exchanged or recombined from one virus to another, creating a rearranged viral genome. So now imagine thousands of pieces of viral DNA or RNA in a cell that is infected by a DNA or RNA virus, and they're having a recombination party. Think about all the unique viruses that can be produced with every infection. I will say that not all recombination results in significant changes in phenotype, but hey, it might, and it could result in a more infective virus. Also, the recombination could lead to just enough change in the capsid proteins so that our immune system couldn't recognize the virus anymore, and that would be bad as well. The second mechanism we're going to talk about is the process of reassortment. This process is a little more straightforward. It is literally when segmented viruses, a virus with more than one segment of DNA or RNA, exchange or trade genetic segments. I know we haven't spoken about this just yet, but what are the 4 segmented viruses? Well, first, they're all RNA viruses, and they're the Bunya virus, orthomyxovirus, arenavirus, and reovirus family. Each circle in this figure represents one influenza virion with 8 RNA segments. Each virus is unique. One has yellow, green, and blue segments. The other has yellow, pink, and black segments. When the viral progeny infect the same cell, imagine all these RNA segments floating around and mixing with each other in the nucleus of the infected, say, human epithelial cell. When the viral progeny finish their protein synthesis and get ready to leave, these different genetic segments or genomic segments can bundle up in unique ways and become an entirely new or entirely unique viral progeny. So here we can see that the segments are actually reassorted from the initial viruses, forming new viruses. This concept, in terms of for influenza, is known as an antigenic shift. This antigenic shift can be very dangerous and can cause the viruses to become extremely infectious and deadly. For example, in the 2009 H1N1 influenza A pandemic, this pandemic emerged after complex viral reassortment of genes from human, swine, and avian viruses. These viruses were very deadly and caused a worldwide scare about the H1N1 influenza. So do you understand why antigenic shift is more deadly and serious? Well, if we remember back to immunology, our body forms an immune response against antigens found on viruses that it has encountered before. So when influenza viruses, like these ones here, undergo reassortment, our immune system is unable to recognize these new viral strains, and therefore they can cause very serious disease. The next process is complementation. Complementation is when genetic material from one virus complements another's. For example, when two viruses such as these co-infects the same cell, virus A can encode a protein that can help out virus B. Here we can see virus B and A, and we can see that although virus B's genetic material didn't change, we can see that it is expressing a new red protein on its surface, which it got from virus A. So here, virus A is complementing virus B. The genotype of virus B stayed the same, but the phenotype changed. We see this process in the hepatitis viruses. Hepatitis D needs an envelope protein from hepatitis B in order to survive. When hepatitis D and B are in the same cell, same liver cell for instance, hepatitis D can steal one of the envelope proteins from hepatitis B and become a happy hepatitis virus. Without hepatitis B complementing hepatitis D would not survive. Now we are brought to phenotypic mixing, the last genetic mechanism we're going to discuss. Phenotypic mixing occurs when the genome of one virus is packaged within the capsid and envelope of another. Let me say that again. Phenotypic mixing occurs when the genome of one virus is packaged within the capsid and the envelope of another. This process results in what is called a pseudovirus. Here we can see, after co-infection into the same cell, that virus A and B switch the capsid and envelope material and vice versa. We can see that capsid or genetic material A, which used to be in the orange envelope and capsid, is now in the purple envelope and capsid, and B is now in the orange envelope and capsid. This is called phenotypic mixing. Now let's say virus A likes to infect the nose, and virus B likes to infect cells of the throat. Take this example. When virus A genes are packaged into virus B proteins, as in this case here, where would this pseudovirus infect? Would it infect the throat or the nose? Well, this pseudovirus would infect the throat because the capsid proteins determine the tropism of the virus. And in this case, the capsid proteins are from virus B. The tropism is a fancy word for where in the body the virus will infect, or what cell in the body the virus will infect. So the key here is that the tropism is determined by the proteins on the capsid of the virus, not by the genetic material on the inside. So although genes of virus A, which are normally the orange envelope and capsid, want to infect the nose, in this case, it is the capsid and envelope proteins of virus B that determine where this virus infects. With this in mind, say the pseudovirus A infects the throat, like virus B would. Will the progeny of this virus resemble virus A or virus B? The progeny will resemble virus A because remember, it is the genotype that is passed on from generation to generation. The only thing that changes in phenotypic mixing is that the genetic material of virus A will infect the area of the body or the cells of the body that is determined by the proteins of virus B because it is in virus B's capsid and envelope. So now time for a flash quiz. To be infectious, on which viral genetic mechanism does hepatitis D virus rely on? Remember, it relies on complementation. Hepatitis B protein complements hepatitis D virus so that it can survive and be infectious.